Emilio Pérez is a doctor that is working at IMDEA Nanoscience in Madrid. That is one center of excellence working on nanoscience. And then he's working in the chemistry of nanostructures, carbon nanostructures, and in particular combining these carbon nanostructures with molecules, with the aim of making some kind of functional materials in which the carbon nanotube is the template or the substrate in which you put the molecule and you try to move the molecule on the top. He will explain this kind of way in which you characterize the system. That is, well, the chemistry is one challenging topic, but also the characterization is another challenging topic. Then what we will learn today will be both aspects, the chemistry part and also the characterization problem of, well, if the system around the nanotube is moving, is not moving, this kind of effects. His topic is, as you see, is inside the topic we are also developing here on molecular materials, on functional molecular materials. Because from the very beginning, he has also been involved in this kind of community in Spain, working on molecular materials through the school and through the European small meetings, etc. Okay, then you can go.
Thank you, Eugenio. I think I'll do this in English, right? There are some...
Yeah.
Right, so I'll do it in English. So thanks for the introduction. I'm actually going to try and tell you 3 stories so that you get a taste of everything that we do now in the group. So one big story is going to be the Rotaxane part, but there's actually going to be 2 others. There's an important part of this first slide, 2 important parts actually. The most important one is this one. So everything I'm going to present here, we do obviously within a team and also with loads of collaborations. So I will name most of the collaborations, but in any case it's important that you know right from the beginning that we don't do anything on our own. Everything we do with many, many people. And the other important part is this one here. We are very happy that we are a Center of Excellence Severo Ochoa since last year. As far as we know, and since we are among friends here, we are the youngest and the smallest institution that has the Severo Ochoa, at least as far as I know. So we are very happy to have that here. Right, so within these many, many others, the most important people are the team. And this is an old picture of the team, but it's a very important one because those are the people that started all the carbon nanotube work. And more importantly, they are people that are already starting to plan their comeback to Spain. So it's important that you know their names and their faces because they will be around, they will be sending emails, and they are very talented people. Alberto de Juan started from the very beginning all the work on the carbon nanotubes. Alex also did a lot of that work. And Leire is also involved in that work. Matthias eventually started with some of the applications, and Leire, I already told you that worked on that too. Mariano and Julia are more involved in the 2D material part that I'm going to tell you about, and Belen was with us for 2 very successful years doing many things, especially related to calculations. So that's an old picture, but I needed to introduce you to Alberto and Alex. The way the group looks now is like this. Actually, we have a new PhD from Murcia, Tomás Nicolás, but the rest of the team also work in similar things. Enrique is our physicist in the team, who's just got an atracción de talento from the Comunidad de Madrid, so he's starting to get independent. So those are probably the most important slides of the presentation. We work on 3 things, and I'm going to try and tell you one story of each. We work on carbon nanotubes, we work on 2D materials, and we are starting to work more and more on very basic supramolecular chemistry, very fundamental questions in supramolecular chemistry. If you wish, this is a little bit our methodology. You know, like when people do total synthesis and they have to develop new reactions, so this is more or less our methodology. So I will try and tell you this story, nearly like the full story. I'm going to tell you one new story which is not published in the supramolecular part, but I think it's very exciting, so I want you to know about it. And then I'll tell you just one short story about the 2D material area. Right, carbon nanotubes. So I think everyone in the audience knows carbon nanotubes, right? Or not really? I think everyone. Otherwise, it's just a rolled up graphene sheet, and they have very, very, very amazing electronic properties. Like for instance, they are like 100 times tougher than steel. They are more conductive than copper, lighter than aluminum. So they are really, really cool materials. but the only thing I usually say on this slide is that they are still really cool materials, right? So they were discovered in the 90s, then graphene came, and it's like carbon nanotubes have disappeared, but they haven't. They are still as cool as they were. Actually, it's probably the best time to work on carbon nanotubes because you can buy really good samples at a reasonable price. So it's actually probably the best time to work on them. So a big part of the research effort on carbon nanotubes has been dedicated to the chemical modification, and the typical reasons behind this is tubes are typically, because they are so long and all atoms are surface atoms, they tend to aggregate a lot, so they are difficult to process into materials. So to improve solubilization, also to improve purification, they are made as mixtures. So you want to control which one of those you want so that you have control over the electronic properties, and also to modify those properties. So basically you want to have access to control the surface properties of the nanotubes, and you want to have access to control the electronic properties of the nanotubes. And as I said, very good time to start playing with carbon nanotubes. Our idea when we submitted this ERC grant that Eugenio was talking about was we were trying to introduce a new tool for the chemical modification of carbon nanotubes. What we wanted to do is introduce this mechanical bond to the chemistry of carbon nanotubes. So before our work, people had been doing covalent chemistry to carbon nanotubes, and this obviously has all the advantages that the covalent bond has, right? So they are very strong bonds. So you have something that is very stable. But it has a very obvious disadvantage, which is you mess up the structure of the carbon nanotube. So all those amazing properties might or might not be there after you do the chemistry, because you're saturating some of the carbon-carbon double bonds. On the other hand, the supramolecular chemistry, the classic supramolecular chemistry, you bring something that the tube likes, typically something that is large and aromatic. So you preserve the structure, that's really good, but you're always subjected to a bound-unbound equilibrium. Regardless of how high your binding constant is, you always have this bound-unbound. So you have kinetically labile products. What we wanted to do is to put rings around the carbon nanotube so that we could combine both advantages, right? So have something that is perfectly kinetically stable, because you would have to break a bond to separate the macrocycle and the tube, but you preserve the structure because there are no covalent bonds between the tube and the macrocycle. So this was our idea. And maybe you're not that familiar with mechanical interlocked molecules, so I'll give a very brief introduction too. These are the kind of things that we are talking about, right? So they are molecules, not supramolecular complexes, because to break them apart you have to break a bond, but that do not have a covalent bonds between the components. So the prototypical examples would be catenanes and rotaxanes. You see the red ring in there is intrinsically linked with the blue ring. So if you want to separate them, you have to break a bond. In rotaxanes, it's the stoppers that prevent the macrocycle from falling, and you can have like even more complicated structures like knots or Borromean rings. And if you like gossip journals, you probably might recognize the Borromeo family in Italy. They are a noble family, like people that are extremely loaded with money. And this is actually their coat of arms, because it's a sign of strength in unity. It's 3 rings that stay together. As soon as you break one ring, the other 2 fall apart. So they are not linked 2 by 2, but only the 3 stay together. All of these have been made and have been crystallized. And funnily enough, around 1 or 2% of the proteins in nature have this kind of structures too. So they are functional in nature. Right, so we do this. This was our idea. Initially, a relatively simple idea, but conceptually, but technically not so obvious, right? So the first thing you have to think is how are you going to try and do this? So if you look at a TEM picture of carbon nanotubes, you will see, as I said before, that they tend to aggregate. So it's going to be very unlikely that you will find the tip of the tube. So if you come with something that is already closed and you try to find the tip, this is most likely not going to happen, right? So we thought it was a much better idea to try and use U-shaped molecules and focus on these parts of the sample where there are single individualized tubes. Okay, that's enough, right? You, let's say, put your molecule in contact with this, you can associate, and then you clip around, then you close it. So that was our idea. And we started with a very simple design where the closing part was going to be double bond, so we're going to use ring closing metathesis, and then we're going to use basically everything that is known to interact with the carbon nanotubes. from the classic pyrene or other polycyclic aromatic hydrocarbons to electron donors like XT3F or porphyrins, electron acceptors, basically anything that is known to interact with carbon nanotubes. Our very complicated reaction scheme is as follows. We put our tubes in a flask, we sonicate them to try and disperse them a little bit, then we mix them with the U-shaped precursors and catalyst and basically wait. After you wait, some of the cycles will be around the tubes, like this one or that one, some others will be outside the tubes, you will have oligomers, you will have monomers, you will have all these kind of things in your sample. And our extremely sophisticated method of purification is we just wash it with a lot of dichloromethane, right? So that everything that is not strongly attached to the tubes is washed away. And even in these, which are cartoons, and which are cartoons that I prepare, I leave impurities. Right? When you work with carbon nanotubes, or in general with nanomaterials, you have to forget about this 100% structural determination. You need to live with this. You have to try and live with this. But you have to make all the effort to make sure that what you're making is mostly this. And luckily enough, our reaction scheme is composed of 2 steps which can be tested individually and from bulk data, right? From very convincing bulk data. The first one is a supramolecular equilibrium, and the other one is actually not an equilibrium. The ring closing metathesis doesn't occur under equilibrium, but it's a unimolecular reaction. So if you test those 2, then you can be sure that your cartoons look more or less like reality. So we spent a lot of time doing these parts in detail. In particular, for the binding constant, we had to develop our own method. People had given up on determining binding constants towards carbon nanotubes, basically because you cannot know the molar concentration of carbon nanotubes. Some of them are longer, some of them are shorter, different diameters, no molar concentration. But actually, if you go back to the textbooks, to the Connors Association constants, this is a book from the 90s, 1970, you realize that all you need to know is this amount. You need to know the concentration of free host, right? So in this, actually, the insolubility of carbon nanotubes helps. Instead of being a problem, it helps a lot. So you just let them equilibrate, you filter once whatever wants to be bound is bound, you filter, and then you measure how much is bound by TTA, and from this you can calculate the free amount. So if you plot these for different concentrations, you get the typical one-to-one binding isotherm, so it works very well. And if you focus on this one, which is the one that matters for this talk, which is one of our U-shapes, you see that the binding constants exist and that they are actually rather high. So they are in the order of 10 to the 4 molar minus 1. And this is work that we've done in collaboration actually with Enrique Ortiz here at IMDEA. Right, so we know the binding constant, the binding part of the equilibrium is true, and it's true for all of the sample, right, or most of the sample. Let's see if this is true too. So for all of you, I don't know how many of you are organic chemists, but those of you who are or know enough about organic chemistry, if you can do this through ring closing metathesis, you can also do that. And you can have oligomers. And then those oligomers might wrap around the carbon nanotubes and bind very strongly, right? So you want to make sure that what is happening last is a closure. And the lucky thing here is that a closure is first order and oligomerization is second order. Right? So you can just do the kinetics and prove that this is a first order reaction, and then you know from bulk data, beyond any reasonable doubt, that most of your sample looks like this. Right? But then what people like to do, so this is the real characterization, but what people like is the pictures. People like to see these things. So we also spend a lot of time in microscopy. So I'll give you 10 seconds to try and find macrocycles there. Anyone? I'm very optimistic, so I see many.
We see one.
You see one? Okay, this one is, let's say, the most obvious, but one could say that this one also looks a little bit like that, like that, like that. So maybe this thing is actually...
They were behind you.
They were behind. Okay. So you can see all these, but I always bring this first picture to show what microscopy is not good for. It is not good if you don't have all the rest. If you don't have all the rest of the characterization, the control experiments, look at the full picture, right? Look at this, and look at all that, and all this. There's no way that you should be convinced about me making Rotaxanes if I don't do the other part. Once that you have one picture where you have like even up to 5, or as you get better, you find many of these objects and they have the right size. As you get even better, you can start seeing the circling around even better. Or if you get to very high resolution, you can even see which part goes up and which part goes down. So you are pretty happy with your synthesis, and finally your referees are also happy, and you can convince people that you've made these mechanically interlocked derivatives of carbon nanotubes. We can now make them with basically all the molecules that I showed you at the beginning. So we can make them with, that first example was with XTF, we can make them with pyrene, we can make them with porphyrins, we can make them with traquinone, so we can make them with anything. This is just to show you how much better we've gotten at microscopy. Now at the Complutense they have an aberration corrected microscope, and we can get pictures that look as pretty as this, right? Where you can see that this is exactly a van der Waals distance, and that if you have a much larger macrocycle like porphyrins, they look like a box, like they should, instead of looking like a ring, like they do when they are, like, let's say, thinner. Right, so is this any use? Is this any good for anything? So when I began, I said, if you want to do chemistry on carbon nanotubes, you either want to control surface properties or electronic properties. One of the most common uses of carbon nanotubes is as fillers for polymers, to reinforce polymers, because they are very strong mechanically. People thought from the beginning that this will be amazing to reinforce polymers, but they are typically very bad at interacting with the polymers, so that the reinforcement falls way below what people had expected. So with this, we thought we had a chance of improving that interface, right? We have something organic around the tube which will prevent its aggregation and looks more a bit like a polymer, so eventually it should help. So we tested pristine tubes, let's say a supramolecular control, and our mechanically interlocked nanotubes. We used 2 different macrocycles, 3 different kind of tubes, so that we know for sure that the effect is due to the mechanical interlocking. We grew some polystyrene fibers through electrospinning, and we basically doped them with some of these materials. And the result is actually very good. They improve the reinforcement a lot. So we get improvements in the Young's modulus of up to 200%, even 240%. We also get a lot of improvement of the tensile strength. We don't get an improvement of the strength to break. Even though it looks like it in some of the graphs, statistically it's not the case, right? So we improve Young's modulus, we improve tensile strength, we don't improve the strength to break. So we know for sure that this has to do with this ideal picture that the macrocycle is helping the polymer interact. And if you run molecular dynamics calculations, and Belen did this, you realize that actually this correlates directly with the mechanical reinforcement. So we know we can access, modulate, and improve the surface properties. So the next thing you want to do is see if you can also modulate the electronic properties. And for this, we thought of another classical function of carbon nanotubes, which is as catalysts. Because all of the atoms of a single wall nanotubes are surface atoms, they are very good as catalysts. They have a very high surface area. And in this particular reaction, which is very simple, it was known from previous work that the role of the carbon nanotube was both as an adsorbent and as an electron reservoir, right? So the final donation of electrons comes from the tube. So this is an ideal playground to see if you can control those electronic properties. If you can make the tube richer in electrons, it should be a better catalyst. If you can make the carbon nanotube poorer in electrons, it should be a worse catalyst. So we synthesized a full collection of these, and we designed one which was supposed to be an electron donor, so make the tube more catalytically active, one that was supposed to be neutral, let's say leave it as it was, and one that was an electron acceptor, so that we would make it a worse catalyst. And again, this is, if you are as optimistic as me in here, this corresponds to this. You can see the full anthraquinone now, and you can even see where the spacer is. You can see atomic detail in the carbon nanotube. So we synthesized this and we tested it. First we characterized it through Raman, and unless you are very interested in carbon nanotubes, I'm not going to go into the details of the Raman characterization, but from calculations we know that the doping effects that we expected are actually there. And if you test this for catalysis, the picture that you get is exactly what you would have anticipated. So The electron acceptor, which are the green lines, is always the worst catalyst. The blue lines, which are the ones decorated with the electron donor, are the best catalyst. And the black and the red lines, which correspond to the pristine tubes and the one modified with pyrene, are more or less the same. So we have access to both surface properties and electronic properties in a way which is not covalent, but is as stable as covalent, and in a way that you can control the degree of functionalization too. So we have, let's say, a very new and potentially useful method for the modification of carbon nanotubes. I think that's that for the carbon nanotube story. I'm just going to wrap up with an anecdote, this part of the lecture. And I think when I explained our reasons to use the U-shapes, was I convincing? Were you convinced when I said that you probably couldn't thread? Kind of, not very. So very recently, this group in Japan has done exactly this thing that I have been presenting for 40 years that couldn't be done, or was very unlikely to happen. They have done exactly that. So they use a very rigid macrocycle, and I think that's the key to their success. And actually it works, the characterization is very solid, and they are, as far as I know, the only other group which are making this kind of things. As you see, the microscopy is also very, very impressive. Right, so that's that for the tubes. Just a little summary. So these mechanically interlocked derivatives of carbon nanotubes are covalent, but the functionalization is as mild as supramolecular functionalization. They have genuinely different physical properties. They do not compare to classic supramolecular compounds or to covalent compounds. You can use them to reinforce polymers or to regulate catalysis. This is something that we have tried. I think the only real application of this eventually might be in sensing, but we still haven't got into that. And didn't talk about endohedral derivatives, but both methods lead you to a modification of physical properties without modification of the covalent structure. And now we're going to completely change fields. So that was one story, the carbon nanotubes. That has been our biggest story in the group, or the one that we have worked more on. And I'm going to introduce a technique that I don't think many of you are familiar with, which are optical tweezers. You have? Okay, so basically in optical tweezers what you have is two lasers very tightly focused. So you use objectives with very high numerical aperture. And these two lights create an electric field there where you can trap dielectric objects. Okay, so in this case, it's a plastic object, typically a polystyrene bead. They are micrometer in size, so microscopic. And you can use this to bring together another sphere with a pipette, actually in a suction in a pipette, make them interact, and then when you pull, this thing behaves exactly as a spring. Right? This is a Hooke's law, so it behaves exactly as a spring, and it allows you to measure very small forces, so in the 0.1 piconewton, and displacements, let's say around the 1 nanometer. So with this you can get very accurate forces, measurements of forces which are just in the range of non-covalent interactions. As you pull, you will measure this delta X or force. So to show you how this actually looks like, so that's where the optical trap is. This is a microfluidic system, so you are always in solvent, and you see that eventually a bead is trapped, and then you come with a micropipette. So this we do in collaboration with Borja Ibarra, by the way. Borja Ibarra is the guy that knows how to use this. You suction that bead, you see that there's a force there already that you're measuring, and then you pump another bead. There you go, it's starting to float around there. It's there if you can see it, trapped. And then you come with your plastic, the one that was suctioned in the pipette. And if you look here, that's your force sensor, and you see as you pull apart, you'll see that that means that there's a force between those two, right? So this is a tool that biophysicists have been working a lot on. And everyone that does optical tweezers was working on biological systems exclusively. So they've used it to study RNA helicases, molecular motors, etc. So like 5 years ago or so, I was trying to convince my colleague Borja of using this tool to study synthetic systems. And eventually, we've started to get results which I think are very interesting, and I wanted to tell you about them. So in this case, for instance, what we are observing is just the rupture of a Hotchkiss complex. So in this case, it's a Hamilton receptor cyanuric acid. We use a lot of DNA tricks to make sure that what we measure is exclusively one Hotchkiss binding event, because these things you can typically do also with AFM, but you don't have access to this unambiguous knowledge that you're breaking just one Hotchkiss complex. So what we use is a double-strand DNA, then something that is called like an unzipping segment, and in here we have 3 markers which are regions rich in GC, which are a bit stronger. So you see as you start pulling, so this will be displacement, this will be force. As you start pulling first, you stretch a little bit this DNA, then you get one peak from the first marker, another peak from the second marker, another peak from the third marker, and then you know when you're here, you're exactly in this position. So anything that comes up comes exclusively from one binding event. So you can measure this, and you can measure the forces which are according to what you should expect for hydrogen bonds, so very low forces in the limit of resolution of other techniques. And in this case, for instance, we also measure that as you vary the pulling rate, you get different forces of rupture. So this means that your system is out of equilibrium. So you get access to a lot of measurements that you couldn't do with other techniques. So we were very excited about this, we were very happy, and we thought we would complicate our lives a lot and try to connect something that much more complicated than a Hotchkiss system. And everyone knows about molecular machines now, especially after the Nobel Prize in chemistry. And this I bring up obviously to give credit to the pioneers, but also one very important message is that mechanically interlocked molecules and molecular machines are not the same. These 2 guys work on mechanically interlocked molecules and machines. This guy works probably on the best molecular machines in the world and doesn't do any mechanical interlocking. He uses different tricks. Right, so what they do is or Stoddard and Sauvage in particular, they make these kind of molecules. If you're very clever, you can devise a way in which you make the green station become the purple station. Then the macrocycle wants to go there, and if you can reverse it, you can make it go back. So those are molecular shuttles, they are called. The real picture, the real physical picture is more like this. So you have a population of these, and they are populating mostly the green station, but they always go and travel to the orange station every now and again, right? Through Brownian motion, through thermal movement. When you reverse it, because that one is lower now, they will start to populate mostly this, right? So this is something that was very well known in the bulk from the 90s. So the first example from Stoddard, he uses an electrostatic approach. So he has a positively charged macrocycle and then a station that he can charge positively with an acid. Then there's repulsion, and the macrocycle goes there, and you can make it reversible, and you can do this with electrochemistry. And these kind of shuttles have gone to the point where you can do useful things with them. So, for instance, this, to me, is my favorite example of a molecular machine based on these things. And this is something that makes peptides in a known sequence. So it does exactly the same function that the ribosome does. So they have gone all this way, knowing very well the bulk, let's say, thermodynamics. But we thought it would be really cool if we could actually measure these things at the single molecule level, right? So that we could understand with atomic precision what is happening or how they work. So luckily enough, I had one of the best PhD students ever, who is Teresa Naranjo, who was able to synthesize this. And for those of you who are organic chemists, again, just follow me a little bit, right? So this here is an oligoethylene chain with 25 oligoethylenes there. So it's basically a polymer. It's no longer a small molecule. So your columns or whatever you're thinking of doing for purification is not exactly the same. This here is click chemistry connected to a DNA molecule of 2,000 base pairs. So this is some serious biochemistry there. And on this other end, we use biotin to attach it to another spacer of DNA. So it looks simple here. It is very difficult to do experimentally. So when we started this project, I thought it would take like 4 years and maybe 2 or 3 PhD students, which I was able to sacrifice because at the time I had my ERC grant, so I could afford it. Anyway, she did it in a couple of years. And when you do this, you can do these force extension curves. So the first thing that happens is that you stretch the DNA a little bit, and this is what the DNA is there for. It's a very known polymer, so you know exactly how it should behave. And then you know that you're pulling only one, right? You know that this is good. If you keep pulling, you make your macrocycle jump, you measure the distance between the stations, and if you come back, you can observe the way back, and you see there's hysteresis there, because actually when the macrocycle gets closer, you get work done from the macrocycle. And this part, I have to say, had already been done with AFM, right? Problem with AFM, you can do maybe 3, 4 of these cycles, and then your system gets fatigued, it breaks apart. With our setup, we can do hundreds. Hundreds, I mean close to 1,000, with no fatigue. And when you can do this, you can get proper statistics, and you can measure a quantity that is very important, which is the coexistence force, right? This is the force where the macrocycle doesn't prefer one station or the other. It doesn't mind. It will go to one or the other. And then you can move to the really, really cool part of this study, which is you set up your instrument to be around the coexistence force, and then you measure the displacement during time only. And what you see is, I think, rather amazing, because you see all the thermally induced motions in real time. So if you look here, the scale is seconds, many seconds, so several minutes, and every one of those is one of those jumps. And if you zoom in, you can see that actually the picture that I showed you, the idealized picture that I drew, is true. So when it goes to the orange station, it actually stays a little bit, and then comes back. And you can see, for the first time, thermally induced motion in real time, and tinker with it, play with it. For instance, you can get the population on both stations, and from that it is immediate to get the energy profile with full detail. And in here we start accessing information that you cannot get from the bulk. Like, for instance, the energy profile does not contain a flat area where the spacer is. It's actually 2 broad wells. You can make it stay on either station equally, or you can make it go to the other station, so you can control everything. By applying the right physical theories, you can extract all the correct thermodynamic data for single molecules, which is something that you cannot access from bulk experiments. For instance, this piece of information is my favorite one, which is that the distance to the transition state is asymmetric. So one would think that those wells would be very narrow, and then once you leave the station, it doesn't matter if you come from that station or the other one, and it does matter. So you have to travel longer distances to leave the fumaramide station than to leave the succinamide. So that's the story that I wanted to tell you about the basic supramolecular chemistry. We are very excited with this, particularly because you can get key information not accessible from bulk experiments, but most importantly because finally, after years of persuasion, there's someone doing optical tweezers with synthetic systems. And this is very important, right? Because now we have all the control of synthetic chemistry and all the detail of single molecule measurements, fully controlled single molecule measurements. So we can get to very deep problems that we thought are going to be very interesting. Like, for instance, we would like to tackle the problem of the nature of non-covalent interactions, or the structural requirements for microscopic reversibility. And I think we will be able to do this in the next 20 years or 15. And one last story, I think I'm fine. It's 30 minutes so far. Everyone still with me? This is the last one, and it won't be very long, so we're okay. So I told you one thing about carbon nanotubes, where I told you basically the full story. One thing about supramolecular chemistry. The other area that we work on is 2D. And here I'm just going to tell you one story that we do in collaboration with the physicists, with the STM people. Graphene is obviously graphene, and everyone loves graphene, and it has all these physical properties which are amazing. But for a chemist, it's also a very nice problem. It's a very nice chemistry problem because all of the carbon atoms in graphene are identical, obviously except the edges. And if you've been talking to organic chemists, or if you are an organic chemist yourself, you'll know that we love to do selective chemistry. We like to do chemistry wherever we want. And well, that's a hell of a challenge. Right? All of the atoms are the same. So, nice problem. Let's try and tackle this. So, already 5 years ago, Ruoff and co-workers published this paper in ChemCom, where what they did was they spread microparticles on a surface, silicon oxide nanoparticles, and then they laid graphene on top, and then wherever the graphene was on a particle, this becomes more curved, and more curved means less sp2, a little bit more sp3, therefore more reactive, and then they did classical Tour chemistry, and they showed that they had some preference for reaction there, right? So I saw that paper, and I have been seeing for many years then at IMDEA, the STM people that grow graphene on ruthenium, they grow it in an ultra-high vacuum chamber, and what happens is because the ruthenium atoms are fatter than the carbon atoms, you get two hexagonal lattices with different parameters, so you get a moire pattern, you get a third hexagonal lattice. And this means that the graphene has basically bumps. So some areas where the carbon atoms are in the middle of ruthenium atoms are elevated, and some other areas where the carbon atoms are directly on top of a ruthenium atoms, those are depressed, right? And this is what they see on the STM. They see a hill and a valley, a hill and a valley. So I had been seeing this, I saw the paper, I thought, let's do the obvious thing and let's say get inspired by Ruoff's work or copy or whatever you want to call that, and try and do this. And this is what happened. So this is the STM images after we tried to do this chemistry, and it was a complete mess. So you cannot even see graphene anymore. It's basically a complete failure. More importantly, even if you don't use your reagent, so if you don't have the Tour reagent, the same thing happens. Okay, and I always like to talk about this example because I think it's one of the examples where you see the advantages of working in a place where people doing very different things are together and work together. Because if I had sent this idea or these samples to someone doing STM in Germany, and the first thing they get is this, that's it. Collaboration finished. Emilio, you're an idiot. Your idea was obviously stupid. And well, they wouldn't write this. They would write, I'm very busy and maybe I won't have time to look at it. But if you work with them, you can go and have coffee with them and try and work why this might have happened. And what happened actually was that the solvent was reacting with the graphene over ruthenium, because the graphene over ruthenium is more reactive than standard graphene. So if you evaporate the solvent very carefully, you can start functionalizing with selectivity, right? So each of those is an attachment. And the selectivity is actually amazingly at the atomic level. So it's only one atom of the graphene that reacts in the unit cell, not the others. The reaction is covalent. Actually, it's the radical of acetonitrile that reacts, and the radical is made in the vacuum chamber as you evaporate it, because the tool they use to measure the pressure is exactly like a mass spec. So you break the acetonitrile molecule. And that's the molecule that reacts, and we know that it reacts through the carbon. This is what how we proved that this species is generated at the gauge, what they use to measure the pressure. So if the gauge is off, there's no functionalization. And this is what my friends, the physicists, call long range. So this is probably a few nanometers square, yeah, 60 times 40. But this, for them, is already long range because it's several unit cells. Obviously, perhaps not long range for a synthetic chemist. But you see, you maintain this selectivity. But if you look down here, 50% is not a very good yield. So if you're a chemist, you want to improve that. So we started playing with the conditions, and if you increase, so this is 600 layers, theoretical layers of acetonitrile. If you increase the amount of reagent to try and make it over 50%, you lose all the selectivity. So not a good idea. So that's the number of layers, that's the temperature, that's the yield. Sorry, the temperature, yield, and selectivity. So if you increase the temperature a little bit, you recover some of that selectivity, same number of layers. If you increase further the temperature, you're starting to get, again, a picture where everything seems sort of ordered. If you increase the temperature and the amount of acetonitrile, you get to 92% yield, 98% selectivity. So you basically are happy with this and finished. And this you see better in like the whole picture like this. And you see that we can get now with very high yield, very high selectivity to functionalize graphene. And there's one big challenge here left, which is to remove this graphene from the ruthenium metal and start doing some electronic games with that. And that were my 3 stories. So I told you a little bit of everything that we do. Those are the conclusions for the 2D part, but I only talked about this part here. So we have a high yielding, extremely selective functionalization of graphene. And that leaves me to thank the team again. So that's the team now. Those are people that passed already through our team, and that should always be there because they've done quite a lot of the work. Our collaborators in the carbon nanotube part, and here we collaborate with Enrique a lot in calculations, although we do some with Angel. We started collaborating in Nazario when we use XTTF. With Dirk Guldi we do photophysics, like everyone that collaborates with Dirk Guldi. Encarna has done some electrochemistry with us. With Dario we started the polymer work, so we didn't know how to electrospin fibers or things like this, so we had to contact someone that knew how to do this. Juanjo is our contact in IMDEA Materiales, so he's the guy that knows that the mechanical characterization that we do is done the proper way, and we use their machines. With David, we do a very similar strategy for the covalent, for the mechanical interlocutor nanotubes, but based on hydrogen bonding. Luisa is our microscopist, so it's not only good to have a good microscope, you have to have a good microscopist, otherwise you don't get those pictures with atomic resolution. So Luisa is responsible for that. Borja is the optical tweezers guy, so he's the guy that actually knows how to measure this and interpret all these things. And these are the people I collaborate with in the 2D part that I told you about. Amadeo and Rodolfo are the STM people, the surface people, and Fernando Martin runs amazing calculations with the whole ruthenium underneath and the graphene on top and all that. And these are the people that used to pay the bills, so that was good because there was loads of money and we could pay all our bills. We pay some of them now from the government, some from Madrid, and quite a lot now from the Severo Ochoa, which has been a lifesaver, at least for my group. And that's all I wanted to tell you. I'm very happy that many of you came, and I hope you enjoyed our little stories on let's say, between chemistry and nanoscience. Thanks very much.
Bueno, questions?
So, I mean, really nice talk. Emilio.
Thanks.
Enjoyed it. I mean, especially because, you know, I'm not an organic chemist, so, I mean, the concepts were really clear. On the strategy for just preparing the mints, I was wondering, actually, this whole process in which you try to pre-catenate somehow and then close the ring, and then you go for the purification in the final step. I was wondering whether if you purify right before, you could somehow just try to control the level of catenation, so you increase the density of catenated material.
Before closing?
Yeah.
This could be a thing. We've never tried that, but we could do it, like filtrating like we do to determine the binding constant. We've never tried that.
Something like that.
But
Because I was wondering, the key limitation of the approach is that it's hard to control the density of catenation, I guess.
We reached the limit. Okay, so this we can do already. So when we finish, let's say, basically the whole nanotube is nearly covered. So that's one of the things why microscopy I know is not very useful, because I know from TGA that we have a cycle every 400, 500 carbon nanotube atoms. So this is nearly fully covered. So we cannot, I don't think we could do it much more functionalized.
And this is more a philosophical question. I mean, compared to the covalent derivatization, which I guess is much more, I don't know, effective in just modifying the electronic structure of the nanotube. I mean, are you expecting the mint just to be complementary to that, in the sense that you have like a finer level of tuning for the electronic properties?
So I think, especially with covalent and with electronic properties, these are one of the properties that go away very easily as soon as you start saturating double bonds. But what I think this strategy is behind, besides being fun, because we've had a lot of fun, is that it will give you access to things that the others don't give you. One thing is in sensing. So carbon nanotubes are very good sensors, but they are so good that they sense anything. Okay, so they are typically very poorly selective. If you have them wrapped around something, and that something recognizes some other thing, that might be useful. Okay, so that's one thing. The really, really fun part scientifically will come from the movement part, which Eugenio was talking about, but we still haven't been able to tackle. Because you have to observe whether these things move or they don't move, and then you get into really nice problems like friction at the submolecular level, and if that energy profile is flat or it's not, and all those kind of things. So that is where I think our approach will pay off.
Thank you.
More questions? Yeah.
In your Rotaxane CNT chemistry, you just always have pre-designed UCM molecule, right?
Yeah.
Which is posed without having any flexibility.
It is very flexible, actually. So I draw them as U-shapes,
Yes.
but they are rather flexible. So if you...
But it's posed, right?
I'll show you the structure. So actually, I'll show you a camera because we will see better with a camera. This is the kind of structures that we make. So we have flexible linkers. Typically, we have an aromatic there, but in this case, we couldn't because otherwise it was very insoluble. And then these are alkyl chains. So it is rather flexible. So these things in solution, they are probably going to be flexible. Then when they meet the carbon nanotubes, this is when they become U-shaped.
And when you are going to react those molecules on the CNT, so can you estimate the distance between two molecules, like two posts?
So as I was telling Carlos, from TGA, we know that we have a macrocycle every 400 or 500 atoms of the tube. The physical distance between them, we can only estimate, we cannot measure yet. Because another problem of TM is that your organic molecules obviously suffer under the E-beam. So you cannot do measurements for minutes or anything like this. You get your micrograph and you go. So we cannot measure it, we can estimate it. They are rather close.
And you have to have that same solubility, right, with the CNT?
So it changes a little bit. Yeah.
It is
So
not, like your post is not soluble with the same solvent with the CNT. How do you manage?
so you typically, so to manage before the functionalization or afterwards, you mean?
Before functional.
We just sonicate like everyone does, right? So it's not fully soluble, but you expand a little bit, you exfoliate some of the tubes. So enough to get some regions to do the chemistry. After the modification, we typically find that the solubility improves a little bit, so you diminish aggregation, but it's not something that we have tried to provoke, like, include solubilizing groups. We have not been interested in that.
Okay, my last question, like in particular Raman, so do you see that there is change in specifically that G-band after wrapping that molecule?
So
Because you have like lot of part coming from that surface form interaction, right? After
So
this molecule.
I'll actually then talk about this because I think this is actually very important. So I assume you are into carbon nanotube chemistry, right? So you like carbon nanotube chemistry, at least a little bit, right? So this is another thing that I've been very worried about because typically on carbon nanotube chemistry, organic chemists, and this might not be good for me to say it because I'm an organic chemist, but we try to extrapolate the results from the physicists which work with a single carbon nanotube and do Raman, and then you find many papers where you see one Raman spectrum, and they say, well, shift it to the right, or shift it to the left, 3 wave numbers, 5 wave numbers, and that with these they extract the conclusions. So if you see, for instance, on the G-band that you were talking about, this is the shift of the G-band, and for any sample, you can find things from 1578 to nearly 1590. Okay, so you can choose if you want to. Luckily enough, 2012, I think it was, there was a group that was studying the Raman on graphene, and they did this work where they separated the mechanical contribution to the shifts from the doping contributions to the shifts. And what they do is they actually analyze the G versus 2D Raman shift. And if you do this statistically, you can see, so in this case, for instance, you can see that the tubes and the one that was not supposed to be doped, they follow along the same line. Okay, so this is mechanical strain. And the ones that are doped are away. Okay, so they are more grouped, they have a different slope. So then you know that your effect is electronic. So we see shifts in the G band. It wouldn't be enough for me to tell that we have doping or we don't have doping.
Thanks.
You're welcome.
More questions? Yeah, Eugenia.
Have you seen any time if one ring is joined to nanocarbons?
No, this is very cool. So we haven't seen this, but we haven't made something that could bind to. Okay, so a molecule that could bind to. And this is something that I think is going to be very interesting to do and that we are trying to do. Because this will help us to make very strong Bucky papers, Bucky papers that people make from tubes, but link them between them. So this is something that we want to do, but we haven't done yet.
I'm curious about the possible application of these functionalized carbon nanotubes for a sensing application. Have you tried something in this direction?
We haven't tried yet, but I think this is the best probability, right? So if you think of having your tube wrapped with something and then a receptor, you could do this, right? If you want to do this properly, what you have to do, I think, is to isolate one tube between two electrodes, then do the chemistry, and then try the sensing. And then you will be able to know if you have improved selectivity or not. We haven't tried in the book because I think it wouldn't give us much information. But I think this is where the possible applications might be, if there are any. Probably there won't be any. The reinforcement of polymer apparently is very interesting. So we have a company in Denmark that wants to make polymers reinforced with these things. Although I told them that I thought it was a stupid idea, but they think it isn't.
Why did you choose polystyrene as a polymer?
That's a very good one. Why would you think? It's a very soft polymer. So it's very easy to reinforce. So we chose to start with the easiest. So it's a polymer that is very weak, and it's also known to be very easily reinforced with tubes. So that is why we chose it. Now we are scanning more polymers, and so far it seems to be a general effect. But we started with the easiest.
Did you observe the influence of chirality on the supramolecular?
Only as far as diameter goes. Okay, so this we see. And this is something that we saw from a spectroscopy with Dirk. That the smaller the tube, the more likely it is to be functionalized. I wouldn't make very big claims in this area. I think it is selective, it's not amazingly selective. For instance, it wouldn't be useful to separate tubes.
And catalysis, it will make also neutrons, probably.
Yeah, it should.
So you have
but for these other examples that I showed you, for instance, in the catalysis part, we buy really, really good samples. So like 95% of them are 6.5.
May I ask you the company?
Yeah, from Aldrich. We just pay a lot.
Okay.
I don't know who makes them for them, but we just pay. I mean, it sounds like a lot, but if you buy 5 grams of this, you can do a thesis. We work on very small amounts, so it's never the limiting factor. I don't know if I should say commercial names. Hi, YouTube.
¿Más preguntas?
No one likes the optical tweezers stuff.
Well, I had one about that, but I was planning to discuss that.
Okay, it's a longer one. Okay.
No, the thing is, you actually need mechanical transport there, like really big movement. I mean, would you?
Not really. So
you could go for something like conformational changes or
so
rotational dynamics of your molecule.
Rotational, I don't think. So in measurement, it's less sensitive than AFM. So AFM would be more sensitive in displacement. Typically, the sensitivity here is in the order of a nanometer. So you need something relatively large.
Yeah.
No, you are an organic chemist. then you don't like to put any metal in your light except some zinc compound. But well, in this kind of systems, in this one, or also for optical tweezers, it's possible to make the same system and attaching very heavy cluster, for example. In this way, you will see much more easy by TM, and also you can move much more easily because you will see them and you can push them optically, for example.
So the limiting factor there is that I don't know my ass from my elbow in terms of inorganic chemistry.
Yeah,
I don't know anything about it,
chemistry,
but I will be very happy to do it.
Some of the systems we have, for example, we are working on the polyoxymetallic chemistry, and polyoxymetallic chemistry allows you to functionalize, to have a cluster, a metal oxide cluster of tungsten that is very good for the temp.
Or even better, in terms of POMs, you can make cycles of POMs. So you could make the whole, can't you?
Yeah, but this is complicated. It's better to take this kind of system and to make decorate in the top the polyoxymetallite, and you have polyoxymetallite on the top, and after you can move much more easy.
So one thing we can do is we can give you something that is functionalized once on the macrocycle.
Yeah,
so that could be a linker to
the POM.
Yeah.
Then once you make with the POM the advantage is that you will see very easy where all the POMs are, and also to move will be much more easy also.
Definitely up for that.
And it's more a hybrid approach with organic, all your organic stuff that you have done is good, but you need, if you add the other part that is not so difficult to functionalize with the other part.
Yeah, that would be really, really cool.
This is a nice system that you can try to explore, because when you will see the contrast is incredible.
And it's also much more stable. So it's the contrast and the stability.
But the contrast is, you don't need a high resolution. With a very low resolution, you will see all the tungsten atoms.
Even on a 200 or...
No, atoms you have 12 or 18, no more.
Yeah, but in the microscope, right? You can see it on a 200 kilovolts, a normal microscope.
With a normal microscope you can see, and it's not destroyed. And I mean, that is very stable with these systems. And this is one thing that we did some TEM with polyoxymetallites, and it's very easy to see the polyoxymetallites on the TEM.
We should talk about that.
And then we did, for a thesis, we did some functionalization with nanotubes with the polyoxymetallite functionalized on the surface. And then we have already nanotubes with polyoxymetallites with a functionalization of the polyoxymetallites to make a kind of we didn't close, but it's the same. But the polyoxymetallite functionalized with organic 2-gram peri-like groups.
We should talk about that. It should be.
This is one possibility for visualizing much more easily and doing much more easily your system.
Yeah.
This is one of the things. ¿Más cuestiones? Bueno, más.
Maybe concerning graphene chemistry last but not least. Did you learn something from the diazolium chemistry experiments you did before the acetonitrile thing?
Mostly we learned that graphene on ruthenium is much more reactive than you would expect. This is the biggest lesson we learned, I think. We didn't learn anything much more than that because everything reacted. And there was no way to control it. Even if you went like very fast, like basically just dip it and take it out, everything
But
reacted.
did you observe a kind of, because with the STM resolution this is good enough, did you observe a fractal growing of the functionalization pattern? Because this is something that the people is discussing now.
We didn't.
So maybe you have to recheck
Go
your
back to
pictures.
that. We have many pictures, so...
Maybe not those, but if you have long range ones, that would be very interesting to check.
So say that again, that we would observe...
So now there are some models claiming that the functionalization is following a fractal pattern with diazolium chemistry because it's very aggressive. So maybe these are perfect experiments to check it. But if you have longer range...
See, we have bigger things. I will... you would have to go through, send me those papers. So you would probably have to do Fourier transform or things like this, or how would you analyze? It's not published.
I can tell you later.
Okay. Nice. So maybe we will learn something. I can do a short summary for the people that have arrived now. Shall I do?
Just the carbon nanotubes, following the approach that Eugenio said on using polyoxymetallites or other interesting molecules. What about getting the carbon nanotube filled with this kind of molecules? Is it affecting the properties? Can you have some multifunctionality or something?
This is something we are doing. I think this is very interesting because it's nice from the synthetic point of view, but it's even nicer from the topological point of view, because actually when something is inside the tube, the topology is the same as when a cycle is around the tube, mathematically. So, you know, in topology, objects are the same if you can make one into the other by pulling them or stretching them, if you don't break anything. So the topology is the same. So we are trying to do both things on the same tube. We are trying to do this in collaboration with Andrei Hlovistov in Nottingham, who's an expert in filling tubes. We have filled some tubes too, but he's an expert. And we are trying to do this.
We are doing the same.
Yeah. You're doing that?
With him.
With him too. yeah, so I saw that in one of your masters.
He worked with Mari Carmen.
He was working with Mari Carmen, but is the person that is doing that. Andrei is the person that is. We have a European project.
So he's the person to work with.
We have done a lot of molybdenum clusters.
Yes.
Some molybdenum clusters that enter in the tube, and you can see the molybdenum organizing several positions.
Have you seen like his latest paper on Nature Communication, where he sees like a catalytic core of atoms in video, like doing all the catalytic stuff to the wall? It's amazing. Andrei is the right guy to do this with. So we're trying to do this, putting something inside and outside. It requires a huge macrocycle, so that was another pain in the neck for one of my PhD students.
For us, it requires a small cluster.
Exactly.
Everything is relative. For us, a small cluster with 3 atoms is enough. With 3 molybdenum atoms, or 3 tungsten atoms is enough. With some personalization around, but you bigger, you cannot fill the nanotubes. No, it's working very well. It's doing some pivots inside there. The pivots we can see. See, like when you put fuel, you have pivots inside, but with the molybdenum clusters, it's possible to see that. Organize in 2 positions, 1 position, and then it's possible to study and also increase the size of the cluster. But it's a lot of things that can be done. The good thing about that is that when you do that, you protect your molecule from the radiation, because when the molecule is inside a nanotube, it's not damaged, but the interaction with the electrons. But if you put outside, the problem you have is that outside your molecules are not surviving for a long time. If you put inside, your molecules are there for.
And also those kind of experiments, because sometimes when I give this talk, like people think I don't like TEM, and I do like TEM. It's just for some things it's very good, for some others it isn't. For these things it's perfect, because you're observing one molecule, and this is what you want, one molecule with detail. You're not doing the chemical characterization, let's say. For this, the things that Andrei does is perfect.
Inside the stabilization.
Yeah, they stay for like in kind of videos.
This molybdenum system are functionalized with organic parts, and organic parts stay for he can irradiate days and days, and it's not changing. Bueno, pues damos las gracias a Emilio.